So I'm just gonna draw out the questions so I don't forget them. So cell cycle, and then checkpoints, and CDK inhib, then damage cell, with activated P53, and a damage cell with mutated P53. And then I'll throw the background information here. So P21 is a CDKI inhibits transition from G1 to S. Okay. And then he binds. Okay. Just leave that up there so don't forget it. Okay. Alright, so a little bit of background information, blah blah blah. So P21 is a CDKI, which is a cyclin dependent kinase inhibitor. So that inhibits the transition from the G1 into S phase. And P53 binds to P21 if it senses damage in the cell. Okay. Alright, so first one is drawing out the cell cycle. So this is made up of 4 different phases. So it has 4 arrows. It's a pretty good circle. Okay, so the first phase, well, one of them is M. Then we have a G1. Then we have an S phase. Then we have a G2 phase. So that's a cell cycle. And like the cell cycle goes from phase to phase due to the checkpoints. And there are 3 checkpoints. There's the G1 going into S checkpoint, so a star there. There's no S going into G2, but there's a G2 going into M. And then there's an M going into G1. So these checkpoints kind of all have different things they're looking for. The G1 checkpoint, we'll start with one pick. So the G1 checkpoint, this one is checking to make sure that the environment is favorable for proliferation. It's also checking that there's enough nutrients and growth factors. And if this checkpoint is satisfied, so if it's a happy one, then it can go along to the S phase. But if it is not satisfied, then it'll stay in the G1 phase. And so the next checkpoint, which is in the G2 phase, so this one is occurring after the S phase. And during the S phase, that's when the DNA is replicated. So this one is asking, is the DNA replicated? If it is it replicated properly? It's also checking to see if there's any DNA damage that needs to be repaired. And if this one's satisfied, this can go along to the M phase. But if it's not satisfied, this one will stay in the G2 phase. So our third checkpoint, which is occurring during the M phase, the M into G1. So the M phase is made up of cytokinesis and mitosis. During this checkpoint, this one's checking to see if the chromosomes are all lined up properly. And then if they're attached to the mitotic spindle, so the chromosomes need to be lined up properly and attached to the mitotic spindle. And so if this one is satisfied, it can go along to anaphase. But if it is not satisfied, this one will stay in metaphase. Okay, so check. All right, CDK inhibitor. So CDK is a cyclin-dependent kinase. A kinase is something that adds a phosphate. So a CDK inhibitor is something that is obviously inhibiting a CDK. Okay. So an example of a CDK inhibitor is P21. And so P21 inhibits the transition from the G1 to S phase. G1 to S. So a CDK inhibitor is something that it controls uncontrolled cell division and can stop or slow down the cell cycle. So stop or slow the cell cycle if it senses damage or like a lack of growth factors. So that can stop or slow down the cell cycle if it senses damage or a lack of growth factors. And that example is P21, which is a CDKI, so CDK inhibitor, which inhibits the transition from the G1 to S phase. Okay. Alright, so in a damaged cell with an activated P53, so what would happen to the cell cycle? So if P53 is activated, that means the cell it senses the damage. And so in that case, the P53 will bind to the P21, and when that binds to the P21, that will make it stop or slow down the cell cycle. And because P21 stops the transition from G1 to S, that would probably be stopped then. So stops in the G1 to S. And then this is known as senescence. For sure spelled that wrong. So in a damaged cell with an activated P53, so P53 is activated due to the damage. The P53 would bind to the P21 because it sensed the damage. The P21 would do its job of stopping or slowing down the cell cycle because it sensed the damage. And because P21 specifically stops it or inhibits the transition from the G1 to S phase, that would probably stop the cell cycle here for this cell. And so that would cause, well, when it can't go from G1 to S, that's known as senescence. Okay. Alright, so in a damaged cell with a mutated P53, so if the P53 is mutated, then it most likely would not bind to the P21, so probably wouldn't bind. Just because it's not working properly. So probably wouldn't bind to the P21, which would not slow down the cell cycle. So wouldn't stop the cell cycle. So this one would keep, like this cell cycle would just keep going, and it'd probably just ignore the checkpoints. So it'd probably ignore checkpoints. And it'd keep just duplicating. So ignore the checkpoints and keep duplicating. And then the damaged cell, so the damaged cells would just keep like amassing, they would just keep going. Damaged cells would amass. I don't know if this how you spell that, but they would just keep duplicating and like add up. And that could eventually cause other mutations or like other cancers to arise. So in this cell, this cell cycle would probably most likely stop because the p53 would sense the damage. It would bind to the p21. The p21 would stop this cell cycle from going in from the it would stop it at G1 going into S, and that is known as senescence. This one, because it's mutated, so the p53 wouldn't be working properly, so it probably wouldn't sense the damage, so it wouldn't bind to the p21, which wouldn't stop the cell cycle. So this cell cycle would probably just keep going, ignore the checkpoints, keep duplicating, and the damaged cells would just add up, and that could lead to mutations and cancers. Yeah. Yep.